Hello guys, welcome to my channel Biological. Today we will learn about the T-dependent B cell activation mechanism. So let's start. So suppose this is a B cell and we also know that B cell has BCR, that means B cell specific receptor on its cell membrane. BCR basically captures a antigen, a foreign antigen that actually enters into our body. After capturing this antigen via the BCR, the antigen is being processed inside the B cell. Actually, there is a specific mechanism of this processing, but we will cover that in another video. After the processing, antigens actually got presented via MHC2 on the B cell surface. This is the MHC2, major histocompatibility complex 2, which is basically present on the B cell membrane. Now, as B cell is presenting this antigen via its MHC2, the T cell comes into the play. So suppose it is a T cell. T cell, like the BCR, T cell also has its own receptor, own specific receptor named as T cell specific receptor, TCR. Actually, TCR recognizes this processed antigen that is present on the MHC2 cell, MHC2 component. Now, the recognition of the processed antigen with the help of TCR gives a signal to the T cell. This is the first signal. Remember that another signal via the co-stimulatory receptors are actually needed for the complete activation of this B cell and also T cell. So what is this co-stimulatory receptor? Actually, B7 receptor is also present on the B cell membrane, which is recognized by CD28, which is also present on the surface of cell membrane of the T cell. It is CD28. So this one actually gives the first signal for the activation purpose, and this co-stimulatory interaction gives the second signal for the activation. Remember, this co-stimulatory interaction is very much important for the activation of B cells. If B7 got mutated, then no activation of B cell can be occurred. So after these two signals, another important interaction occurs between this B cell and T cell. via CD40 and CD40 ligand. The importance of this interaction will be told later in this video shortly. So let me draw this CD40 and CD40L. This is the CD40 and this is CD40L. CD40L is present on the T cell and CD40 is present on the B cell. So basically these three types of interaction occurs. After these interactions, basically after these two interactions or two signals, the T cell got activated. So what will be done after the activation of T cells? Actually, after the activation of T cells secretes some cytokines. So what are the major role of these cytokines? Actually, these cytokines got recognized via a receptor called IL-2 on the cell surface of B cell. This is IL-2. So these cytokines actually got recognized via IL-2 receptors which are present on the B cell surface. After this interaction, actually B cell got activated and the differentiation of B cell to the plasma cells occurs. Plasma cells are basically the antibody secreting B cells. So when a antigen enters, foreign antigens actually enters into the body, so in this way the B cell got activated and via the T cell, and then after the activation B cell actually differentiates into the plasma cells, and plasma cells basically secretes antibodies to counter the pathogen. So by this way the activation of B cell via the T cell actually occurs. Antibody secreting plasma cells via these T dependent B cell activation mechanisms. And one thing is important, that from these plasma cells the first class of antibodies that are actually produced are immunoglobulin M. Always immunoglobulin M is produced at first from these differentiated plasma cells. Now there is a question: if immunoglobulin M is being produced from the differentiated plasma cells via these interactions, so how the others antibodies can be produced? Actually there is a mechanism which is called class switching is present. It is known as class switching. Also it can be called as isotype switching also. Isotype switching. So via these class switching mechanisms, actually different immunoglobulins or antibodies like immunoglobulin A or immunoglobulin E, G can be produced. So this is very important to remember. And besides this, another important mechanism is also present, and actually I will discuss about both of these mechanisms later. Another important mechanism is also there to ensure proper action against the foreign pathogen, which is known as somatic hypermutation. Somatic hypermutation. So basically, what is somatic hypermutation? I will talk about it later. Let's just allow me to focus on this particular topic, which I am discussing in this video. So as I have told earlier that the interaction of CD40 and CD40L ligand between the B cell and T cell has an importance. So what is the importance actually? Actually, the interaction of CD40 ligand and, so just let me draw CD40 ligand, which is basically present on the T cell surface, and the interaction of and the CD40 which is present on the B cell surface. Actually, this interaction causes activation of these two important mechanisms. Remember, if any of this CD40 or CD40L got mutated or this interaction can be occurred, then these two important mechanisms, which is class switching and the somatic hypermutation, can't be occurred, and it would be very fatal. So our discussion ends here. This is a very general outline of this T-dependent B cell activation mechanism.